LiDAR maps reveal unprecedented details about the bare Earth, as well as the patterns of the trees and shrubs that cover the landscape. Scientists from the National Center for Earth Surface Dynamics, NCED, based at the University of Minnesota, are using this data to explore the interplay between the topography and the animals and plants that live on the land. How fast can a stream current flow before underwater grazers are washed away? How does the availability of sunlight change the river food web in different parts of the watershed? Where do threatened steelhead get nutrients in the dark headwater streams? Predictive mapping, using topographic data to predict environmental regimes, is helping scientists find answers to these questions. Mary Power is a professor of integrative biology at UC Berkeley.
We are taking advantage of the technology and the mapping skills that they've provided to follow food web interactions up the drainage network, moving from sunny mainstem, warm, productive habitats upstream where it's cooler and darker but tends to stay more moist. Organisms like lizards that are favored in hot, sunny environments become slowed down and less important in the dark habitats, and organisms like spiders that are abundant and important in the dark, moist habitats start to desiccate and drop out in the dry habitat. So you can have the same characters in the food web, but their relative importance and the strengths of their interactions change. And so in our attempts to take advantage of earth science for predictive ecological mapping, we're looking at the thresholds for where an ecological regime changes. Where can a grazer like a tadpole or a grazing caddisfly control algae, and where is its presence irrelevant to whether algae accrues or not? Where can bats effectively track emerging insects like mosquitoes, and where is the drainage suddenly so steep that the white noise jams bat ultrasound and they become ineffective? Angelo Reserve is a fantastic place to study predictive mapping because we have such steep gradients that occur over short distances. We can get to them all in a day or 2 days. We can study drainages that are a half a square kilometer and just maybe 10 inches wide to this drainage here, which is the main stem South Fork eel that has an active winter channel of about 30 meters and a summer channel of about 20 meters wide. So we tend to repeat the same experiments, whether it's studies of alders, of caddisflies grazing algae, of bats tracking insect emergence. We're repeating those experiments at the same 8 or 9 watersheds to get contrasts, to see where there are breaks in ecological outcomes with drainage area, and then trying to interpret why they occurred, and with that, hoping to find ourselves in a position to predict what would happen in the future were environmental conditions to shift around because of climate change, land use, or biology. That's the philosophy of predictive mapping.
LiDAR mapping isn't the only advanced technology being used by the NCED team. They're also using stable isotopes to track minute changes in oxygen, nitrogen, and carbon atoms that reveal an animal's food sources. Jacques Finley, an associate professor of ecology at the University of Minnesota, is leading this effort.
Stable isotopes are one very useful tool that we've been able to make a lot of progress in terms of mapping out feeding relationships between organisms. Some stable isotopes act as conservative tracers, so once they get into a food web, we can follow them through the food web and actually see how carbon, organic matter, is moving through the food web on up to top predators, salmon and steelhead.
This NCED collecting team is making its way to a headwater stream in the reserve to collect samples for a study on how the steelhead diet varies across the watershed. Camille McNeely, an ecologist from Eastern Washington University, heads the team.
One thing that's important is we never want to mix riffle and pool bugs. So some of these bugs, like the mayflies and the stoneflies, will find both in pools and riffles. And we just want to have a separate vial for the ones from the pools.
We're trying to understand what fish eat in this watershed. And this goes back to some of Jacques Finley's work from his dissertation, where he found a pattern where there's a really sharp transition in the watershed between predators, including fish and also invertebrate predators that use terrestrial carbon. Somehow it makes its way through the food web once it falls in the stream, and they're supported by terrestrial carbon in small streams, algal carbon in large streams, and there seems to be a fairly sharp transition at about 10 square kilometer drainage area. And so what we're interested in doing now is trying to explain that pattern. I mean, it makes sense that there's a transition, but why does it happen where it does, and why is it so sharp? I guess what really interests me is not just explaining that transition, but we're talking about predators and the energy that sustains them. It's being fixed by plants, either terrestrial plants or primary producers in the stream, algae. And they're not eating plants, they're eating something else. And so I'm interested in the pathways through which that carbon and energy is moving through the food web to get to the predators and how that changes.
One possibility is that they're feeding on terrestrial bugs that just fall directly into the stream, and then at some point maybe those are not so abundant as the canopy gets wider over the streams. Another possibility is that they're feeding on bugs that live and grow up in the stream but that are sustained by detrital inputs themselves. And then another possibility is that there's dissolved organic carbon that comes into the streams as groundwater and that leaches out of leaves and stuff like that. And that supports slime on rocks, which we might superficially expect to be algae, but actually in a lot of the streams, a lot of it is bacterial. So it may be that bugs are feeding on that, and then fish are feeding on those bugs.
All right, fish folk, are we organizing down here?
One group focuses on finding steelhead.
Oh, that's it, I can't.
They capture the fish with a quick electric shock, sedate them, and flush out the contents of their digestive systems with water. Then they weigh and measure each fish before returning it unharmed to the stream.
Standard length, 112.
24.96.
Meanwhile, another group is collecting live insects that can be matched isotopically with their partially digested colleagues.
When we look at the bugs, we'll see if they're terrestrial bugs, if they're aquatic bugs. What do we know about the diets of those aquatic bugs? We've actually done quite a bit of pretty intensive work on the diets of some of these. And so we know which bugs the fish are eating, then we'll know, are they biofilm feeders, are they detritus feeders, are they algivores, etc.
Throughout an animal's life, its diet is recorded isotopically in its body tissue. Because terrestrial plants and aquatic algae have distinct isotopic fingerprints, the scientists hope they will be able to trace the steelhead's food energy source all the way back to the point where the carbon was fixed in the environment. After a long day of collecting, the team's work is still not done. In the evening, they return to the reserve's laboratory, where they work late into the night, sorting and preparing the specimens they've collected, removing their protective cases, and placing them in clean water overnight to flush out their digestive tract.
To get good data on isotopes and nutrient content, we really just want to know what the bug tissue is. We're not interested in their last meal, and so we're going to keep them overnight in the lab and hopefully allow all the material they've eaten to pass through their system. We're trying to understand some basic aspects of food webs, how fish that are in fact listed as threatened feed themselves and survive in these streams.